One more week, more or less. Then no more virology. Sniff. So last couple of lectures are really going to be kind of different from what we've had before. These are going to be very much more overview lectures. You could do whole terms on plant viruses, and we're going to do one lecture on them. You could do whole terms on archaeal viruses. In fact, that was the meeting that I was at last week, was all about archaeal viruses and archaea in general. And then the viruses of algae, some of the giant viruses, again, you could do a whole course on those. So these are sort of much more overview kinds of things that we're going to be doing for the next couple of lectures. The other thing is, yes, there will be clicker questions today, but as I was putting them together, I realized I didn't put together any clicker questions for the adenoviruses as well. Would you like clicker questions for those? Yes, no. Yes. Do we care? Yes? No? Yeah, just click on it. I know I should do the plus minus click. So the problem is that would screw up the order, and I'm already having trouble trying to get all the clicker scores posted, so I'm trying not to do that. Okay, so it looks like you would, so I'll just have double clicker questions next time. Okay. That work? Good. Okay, so that being said, our clicker questions for today, which are on those other viruses that we talked about last time. Parvoviruses use blank for genome replication, protein priming, RNA priming, DNA priming, A and C, A and B. $30. $10, $9, $8. So yes, it is DNA priming. Yay. Protein priming is certainly true for a lot of the other DNA viruses that we talked about, and also some RNA viruses. RNA priming, of course, what happens normally cellular replication, and it turns out if you're talking about some of the other single-stranded DNA viruses, they also require RNA priming, but not these parvoviruses. And then, so it's just the DNA priming. So that would be this one. Our next question is, adeno-associated viruses depend on what kinds of proteins from adenoviruses? Again, feel free to discuss these things. I don't. 8, 7, 6. So, capsid. Nobody seemed to like capsid. I'll show this in just a second. I don't want to block the whole thing here. Turns out there are some really fascinating both bacterial and archaeal viruses that use a capsid protein from an associated virus to do that, but not adeno-associated viruses. They actually have their own capsid protein. Host modification, yeah, that's what the answer is. DNA polymerases, and that's what E1A does, among other things. The DNA polymerases, what DNA polymerases these guys use? Adeno-associated viruses? These cellular polymerases, exactly. Priming enzymes. They don't need any priming enzymes because they use their own DNA polymerases. But also this would be the protein priming, potentially, that you would have for the adenovirus. And endonucleases?
Probably cellular.
You think?
That's what you actually said in lecture.
Yes. When you think about the integration, however, for replication, what do they need? What's the endonuclease they use for replication?
My jaw hits the floor.
So what endonuclease do they use for replication? Yeah, it's the adeno-associated virus. It's the rep protein or the NS1 protein, which it uses to nick across from the end to provide the primer that it's using for replication. So yes, it is B. Okay, last clicker question for today. My favorite virus, well, almost my favorite virus. The RNA-DNA hybrid virus is most similar to blank in its genome form. AAV2, polyomavirus, FIX174, adenovirus, or HIV1. D&E is not a possibility. No, F doesn't. Looking for the button on the bottom of your clicker. The F button. Time to start guessing. So, what form does the RNA-DNA hybrid virus have? This genome form, what is it? So, well, okay, I heard something there. It just went a little too fast.
I said, please say circular overlapping.
Please say circular with overlapping reading frames. Overlapping reading frames is incorrect. Circular, however, is correct. But is it circular double-stranded DNA, circular single-stranded DNA, circular double-stranded RNA, circular single-stranded RNA? Circular single-stranded DNA, which is similar to FIX174. So, single-stranded DNA, like AAV2, double-stranded circular, which would be the polyomaviruses, adenovirus, linear, double-stranded, and HIV1 is what? So what's the genome that's in your HIV1? Single-stranded RNA. But these guys probably stole an RNA from a co-infecting virus. Exe und omnes parado ad astra. How many people had Latin in school? Exeunt omnes is everybody leave. So, okay, so today we're going to talk, again, at a very peripheral, 37,000 foot level view of plant viruses, and particularly the RNA viruses that are infecting plants. We talked a little bit about the DNA viruses, particularly the single-stranded DNA viruses that infect plants last time. What were those called? Gemini viruses, exactly. So the Gemini viruses are the single-stranded DNA viruses which are infecting plants. Today, we're talking about the RNA viruses. I thought about bringing that ridiculously ginormous book, the virus taxonomy book. About 2/3 of that is plant viruses. So it gives you an idea how much simplification is happening here. Just wanted to mention the way that a lot of these plant viruses are named. It's basically the name of the plant followed by the phenotype of the disease. So in this case, bromovirus is the Mosaic virus, which gets really complicated and confusing, particularly since a lot of these viruses infect a whole bunch of different plants, but at least the first one that was found. Many of these guys have segmented genomes, which is really curious, particularly since those segments actually often end up in different capsids, and that has to do with how the infection process takes place in these plant viruses. Obviously it works, otherwise you wouldn't have so many of this particular form. Probably mostly has to do with the fact that plant defense systems are mostly having to do with protecting the plant from the outside because it's hard for plants to run away. So here, that's one aspect. The other thing is that once you get inside a plant, it's actually hard to get from cell to cell or plant to plant. And so very often what you need are these so-called movement proteins, which again are very specific to plant viruses. Again, key concepts here, at least now, these are the RNA viruses. There are, again, quite a few of particularly the single-stranded DNA viruses that are infecting plants. These segmented genomes. What other kind of segmented genomes have we talked about so far? Influenza virus, exactly. So it has a segmented genome. But those are all packaged inside a single capsid, whereas in most of these RNA viruses of plants, the segmented genomes, they're packaged in multiple capsids, which is bizarre, at least as far as I'm concerned. But again, shows evolution is something that works. And again, these are plants. One thing that's really interesting about these virions, and we'll talk about one in particular, is that very often there's a conformational change that happens on the virion, but that stays actually very, very stable. And a lot of these plant viruses are extremely stable. And we'll talk about why this is very useful, in fact, for some biotechnology aspects of things. Some of the effects on the host, again, it's most has to do with the movement proteins. And then these things, again, are incredibly resistant, heat stable, etc. And then finally, at the end, we'll talk a little bit about some helical structures. The book chapter, which I've assigned for this time, is on CucumberMosaic virus. Turns out that CucumberMosaic virus infects almost all plants, not just cucumbers, but originally discovered in cucumbers. And again, we'll do our pretty standard process here. Where did it come from? What's its structure? The structure is a very simple T = 3 quasi-equivalent icosahedra. Entry and transmission, this is probably the main thing which is different again with all of these plant viruses, that you have to have a very specific way of getting multiple virions inside the cell in order to get relatively decent replication. Packaging, again, is multiple different ones. I split up this set over here because these are really much more general and separate kinds of things. Satellites, these are small RNAs that are often associated with these RNA viruses of plants, some of which make a huge difference in terms of the pathogenesis, and we don't really quite understand why. Some of these things are just tens of nucleotides in length, but they still have a huge effect on pathogenicity, and again, quite why that is not entirely clear. Talk a little bit about CCMV. This is chlorotic mosaic virus. Because it's been probably the best studied in terms of having this really stable structure and being used for biotechnology. In fact, people are using CCMV to try and make ultra-high density hard drives, which is really kind of bizarre that a plant virus that you're using for real crossover here. Hot viruses, there's a really great story of some plant viruses, in fact, fungal viruses that are infecting plants in terms of giving them ways that they can survive at very high temperatures. Again, quite how that works is not clear. And then again, a virus we could spend the whole term talking about, CucumberMosaic virus towards the end. So this is basically the outline of what we're going to be talking about. Just as a reminder, again, these are single-stranded RNA viruses, positive strand. In order to, of course, replicate, then you need a viral RNA-dependent RNA polymerase, but you don't have to code that. You don't have to be bringing it with you inside the virion. It's just something that has to be encoded for in the virus genome. So where does CucumberMosaic virus come from? Basically, you find cucumbers that have this strange phenotype, basically blotchy coloration that you see on leaves, you see on the cucumbers themselves. Was found very, very early. In fact, 100 years of CucumberMosaic virus will be 2 years from now. I don't know if they're having a big party. We shall see. They're having a big party about bacteriophage next year. So these are, again, the cucumoviruses, CucumberMosaic viruses. Again, don't ask, just historical. That's how a lot of these plant viruses were named. And it's not just cucumber. Infects tobacco, infects trees, infects, sort of you name it in terms of different kinds of potential hosts here. And this is just a one site. You go online, you can find infinite numbers of pictures of what the phenotype of these look like. Turns out it's really variable, and this is probably why the virus has been so successful, is it doesn't, in fact, kill off the plants themselves. It just gives them a slightly off phenotype, at least as far as we're concerned. But it's not killing them off, and so that's probably why it actually is maintained in the population, in all these different populations as well. Quite why it has such a broad host range is not completely clear at this time. The structure of one of the capsids, very regular T = 3 icosahedron. And let's see, we can do our fun review now on what these structures look like. Come on, where's my mouse? So T = 3, hopefully should be really obvious where this is. What do you need to do to start with? Find a 5-fold axis of symmetry. So here's a nice 5-fold axis of symmetry right here. Where's your next 5-fold axis of symmetry? Here's a nice one down here. And now you just need to go from this one to that one via wherever your hexamers are. So you take a single hop here. That's an h is equal to 1. Change directions. K = 1. 1 squared plus 1 times 1 plus 1 squared is a T = 3. It's all made of one capsid protein. So it's that, again, completely quasi-equivalent structures. So these proteins can either form pentamers or hexamers. When I was reviewing for class today, I noticed that they mention this arginine-rich part of the structure, this helix down here. Did anybody else notice the typo in the text? What's the charge of arginine at neutral pH? Arginines and lysines have positive charges. They're likely to be interacting with what? Negative charges, which you have in your DNA, yes, but are these things packaging DNA? No, they're RNA, so it's interacting with the RNA. I think if you look in the textbook, you'll see it says, this whole stretch of arginines, the negative charge of arginines interacting with the nucleic acid. Like, huh? So, yes, it's clearly a typo. So this arginine-rich helix here in the middle of the capsid is clearly interacting with the RNA. And then you have this beta barrel structure on the outside. Beta barrels, again, we've talked about many times now in terms of structures of icosahedral capsids. Talked about it for STIV, but the big difference with STIV and adenovirus, and as we'll see for some of the giant viruses, those have beta barrels that are kind of sticking up relative to the outside of the capsid, and here the beta barrel is really very flat relative to the outside of the cell. And this, in fact, looks very similar to poliovirus in terms of the structure, how it's actually put together, although the sequence of the protein is really very different indeed. So these are the capsids, and this is, in fact, a Plant Virus Institute down here. So again, one lecture versus a whole institute. How do these viruses get into the plants? Almost always it's through a hole in the plant, because plants have really good defense systems. Basically, just the outside of the plant is really resistant to herbivores, et cetera, et cetera, et cetera. So the idea here is that the way to get your virus inside the cell is to make a hole in the cell. And that hole can either be made by our friends here, the aphids, or just by plant damage. And so most plant virologists, when they're doing infections, they'll just scrape the top of the leaf and then pour some virus on it. It's a pretty standard way of doing an infection. But if you don't have any damage to the plant, you could put 10 to the 9th virus particles on the outside of the plant, sitting on a leaf, nothing would happen. And so that's, you have to have this break in the outside shell of the plant and the plant cell wall in order to get these guys in. But once you've made that hole, those are really usually very, very big relative to the size of these virus particles. Now, they're about 30 nanometers. They're really very, very small. So that big hole, if you're making a big hole and you've got a whole bunch of virus, then you can get away with multiple capsids coming in at the same time. And so it turns out that the multiple capsids are all encasing just one of the multiple segments. So each of those segments is in a separate capsid. And the only reason that this works, because if you're thinking about this from an aerosol transmitted or something like that, where you have very few virus particles, like most of the animal viruses we've talked about so far, that's not going to work. Bacterial viruses usually have a very specific way of getting through that cell wall. And so those are the single ones. But here, you've got a friend helping you out, the virus friend here, our aphid, making a hole, and that thereby being able to introduce lots and lots of virus particles. Now, it turns out that the CucumberMosaic virus, probably part of the reason why it's so good, can survive very well inside the aphid. And so it's sort of a part of an aphid life cycle. And people like to think of this as kind of an analog for some of the arboviruses, the insect-transmitted viruses that are infecting humans, that can then also survive inside some of these insects. And so people are trying to make some parallels between the two. So makes a hole, injects the viruses, the virions in there, and then what's in those. So there are multiple capsids, and this is true for CucumberMosaic virus, but many of the other plant viruses as well. You've got multiple RNAs, again, segmented genomes, each of them packaged in a separate virion. In this case, for CucumberMosaic virus, RNA1, RNA2, and then RNA3 and RNA4 are packaged. One of the things that seems to be the way that you get these T=3 particles packaged, again, all at the same size, is you've got about 3,000 nucleotides that gets packaged inside each of those particles. So RNA1 is about 3,000 nucleotides, RNA2 is 3,000 nucleotides, RNA3 is only 2,000 nucleotides. To get a full particle, then you need RNA4 and RNA3. And RNA4 is just a subgenomic piece of RNA3. So a subgenomic RNA. So we've already talked about subgenomic RNAs for coronaviruses. You also have them for retroviruses. So it's not an unusual process. These RNAs that come in all have 5' cap structures. So they're ready to be translated by the cellular machinery. And they've got really curious structures at the 3' ends. Almost all of the plant virus genomes, at least the RNA genomes, have tRNA-like structures at their 3' ends of the genomes. So we'll take a look at those now. This is just one example of one of these structures here at the end of, if I remember correctly, this is RNA1 of CucumberMosaic virus. Has a particular RNA, folds up in this structure down here at the 3' end of the genome. This structure, of course, doesn't look to me like a tRNA, but if you look at it in 3 dimensions, and what's looking at it in 3 dimensions, of course, are all the cellular proteins that are normally involved with tRNAs. So the CCA enzyme, which adds CCA to the 3' end of most of the tRNAs will recognize this and add usually just an A residue, but in sometimes all CCA. It depends again on the plant virus. Also, this looks enough like a tRNA that the aminoacyl tRNA synthetases will bind to this structure and even put an amino acid on the end. And so normally it's the 3' end where you get the modification, or aminoacyl tRNA synthetase that then gets to be used for translation. Once you have a amino acid hooked to the 3' end, this seems to serve to protect that 3' end from the nasty exonucleases which are floating around inside the cell. And so you think about 3' end, you've got your cap structure, which is important for getting translation to work, but also important for protecting against exonuclease activity. Same thing is true here at the 3' end. So this is protecting against exonuclease degradation of the genome. So that's what the ends of the genome again. You've got a cap structure, you've got an end structure. Where do those caps come from? Any ideas? They don't go to the nucleus and get snatched. So as is true for most of the other capped messenger RNAs that we've talked about, with the exception of influenza, these guys make their own capping enzymes. And so if you look at what proteins are encoded in the various different RNAs, RNA1 has a capping structure. I say the protein encoded by RNA1 has capping activity. The other thing that these guys absolutely need is what? RNA-dependent RNA polymerases. That's encoded for in RNA2. What else do you need to make virions? Capsid protein. And so that's in RNA3 and RNA4. Mostly RNA4. And again, arginine-rich domain at the N-terminal region, important for RNA binding. That's the basic capsid protein. A lot of people talk about these as the CP proteins. And in fact, it was the CP protein that we found when we were looking at RDHV. We're saying, okay, it's a CP protein that actually looks really similar to some of these RNA virus-like capsid proteins, but it was in a DNA virus. And that was the whole bizarre process that we found got whole started with this in the first place. There's one other thing that I wanted to mention here in terms of the virus proteins. So we've got capping proteins, we've got our RNA-dependent RNA polymerase and capsid. The only other protein that's needed here are the so-called movement proteins. And these are specific for plant viruses, and they're ways for getting viruses to move from one cell to another. And this is really critical, again, in plants, because if you've infected one particular cell, because that's where the aphid happens to have bit, if you want to have transmission of those particular viruses, they've got to get to other cells in the body. And that process usually is difficult because plants do control quite well how they different infections, to some extent, get from one part of the plant to the rest. And so these movement proteins are really important for getting the virus, and sometimes even the whole virion, from one cell to the next, and often, in fact, through the circulatory system of the plant, the vascular system of the plant. But before that, you need to replicate your genome. How does that work? Again, these are the viral encoded proteins. Turns out that capping enzyme is also part of the RNA-dependent RNA polymerase. If you just look at the top here, that would be one of those stereotypical RNA genomes. Have structures at the 5' end that I haven't talked about yet. These are actually downstream of those cap structures, almost always a stem loop structure. And that stem loop structure seems to be a very critical site for binding to the capping enzyme, which again makes sense, so that's where you're going to be wanting to put the cap on, but also serves as the place to say, hey, this is where we want to be making our negative strand genome, because again, these are all positive strand viruses, and then use that as a nucleation site to get these viruses to replicate. Another thing that we know about a lot of these plant viruses is they replicate associated with membranes. This should sound really familiar relative to things like the picoRNA viruses, the flaviviruses, etc. They all like to replicate at membrane structures, and that's probably just because it allows you to get all the proteins together that you need to get replication to take place and concentrate them in some particular places. So here you have binding of the capping enzyme and the RNA polymerase, make these membrane vesicles, and basically crank out RNAs. In some cases, these are then exported from these vesicles. Some people think, although this is still kind of controversial, one of the reasons to make these sort of vesicles is to protect the double-stranded RNA, which you automatically have as an intermediate for replication, from the RNA interference machinery. And RNA interference was originally discovered in plants. And so plants have a really good way of defending against double-stranded RNA, probably because of virus infection. And this sequestration of these double-stranded RNAs away from the rest of that RNA interference system is probably a way that these viruses use to replicate. There are also cellular translation proteins, particularly EAF3, that are involved in replication. And again, this should sound really familiar based on a lot of the other RNA viruses that we've talked about before. These are proteins that bind really well to specific structures in RNAs and then help to get your replication to take place. There are some folding enzymes that also seem to be important for this process. In fact, some of the cellular proteins seem to be replaced by some of the viral proteins in this process. Again, it's just folding enzymes putting things together. I mentioned movement proteins. These are now proteins that help get either the virus genomes or, again, in some cases, in fact, whole virions moving through the whole plant. So this is just a cartoon of the plant up here at the top. 2 plant cells connected by plasmodesmata, which are usually tightly closed. These virus movement proteins will often bind to proteins that are present at the plasmodesmata and help those to open. And so that's the way you can either get, again, transmission of genome, or in some cases, transmission of whole virions from one plant cell to another. And if you get this transmission to the appropriate vascular part of the plant, then you can get this systemic infection. So you've got one particular site, again, where the aphid bit and injected all of its capsids. Now that can spread throughout the whole plant. And then if people, one of the ways that we know this, if you have mutations in particular plant virus genes where you have mutated these movement proteins, you'll have a local infection. And so that local infection will stay basically right on the leaf or wherever it is that you did the inoculation. However, if those movement proteins are active, then you can get the spread of that infection throughout the whole plant. So that's the process which is going on there. One of the things that I didn't put up a slide for here, but I did want to mention, I'll actually try and bring one up next time. One of the ways that plants actually try and deal with viral infections is to block that movement. And so they'll sacrifice a leaf or some portion of a leaf in order to not have a systemic infection take place. It's called the hypersensitive response. And so if you infect one particular leaf, what will happen is basically an apoptosis process whereby all the cells around that individual infection will die, but the rest of the plant will be fine. And so there also are plant mechanisms for dealing with these movement proteins, again, in the same particular way. So that's CucumberMosaic virus. Again, very wide host range. People say it's the widest host range of any virus. I could argue with them, but clearly plant virus people wrote this chapter. Structure is very regular, T equals 3 icosahedra. The really fascinating thing is you've got multiple of these virions, and you have to have infection by multiple virions in order to get productive infection to take place. And that's only works if you have some kind of mechanical mechanism and a big hole, basically, that you make in the plant cell wall in order to get these virions in. And so transmission is almost always going to be vectored in some way. Replication, It's a pretty standard RNA-dependent RNA polymerase process. The differences are those things that you have at the ends, again, the cap, which is a virus-encoded capping enzymes, and then the 3' end, which is a tRNA-like structure. Packaging is that not negatively charged arginine N-terminus, but positively charged arginine N-terminus on the capsid protein, which seems to encapsulate the 3,000 approximately nucleotides that you have on any of those given RNAs. So any questions about the sort of general process here of these cucumber mosaic viruses, again, is sort of the paradigm for plant viruses, which is not a terribly good one. Yeah?
How does entry into the vascular system of plants cause a systemic infection if you need a hole initially to get the plant virus into the cells?
Oh, so the question is, how can you get basically from the vascular system back into a replicative cell? That's an interesting question, and I think it probably also depends on movement proteins. Getting then presumably the genomes, and again, now sometimes you'll have the capsids which are being moved with these movement proteins, and sometimes it'll be the genomes which are being moved by the movement proteins. But presumably you have to have a pretty high infection load to be able to do that, because again, you've got to have all three, in the case of cucumber mosaic virus, of those capsids or RNAs for that matter in a particular cell in order to get good replication. So you have to have a pretty major infection, and that's probably why the hypersensitive response works reasonably well. And so there's a certain period of time that you have to then isolate and contain that infection. Do you have a question? Yeah.
Yeah, because you're packaging all the genomes in different particles, do you see a lot of reassortment like with influenza or something like that?
So you don't. So the question is, do you see reassortment? Basically, because you've got that 3,000 nucleotide limit on any one of the individual capsids, that's going to be pretty selective. And so sometimes you'll have a whole bunch of capsids that just have 1, or just have 2, or just have 3 and 4. But in order to have a productive infection, you've got to have 1, 2, 3, and 4. So you don't really see too much in the terms of reassortment. You don't see the recombination that would be happening inside one of those individual genomes. Yeah?
So was it the aphids that are also carrying the virus, or how does the virus move around from one plant?
Yes, the aphids carry the virus. And so it seems that when an aphid takes a meal, and then probably also from the vascular system, because they're often going to be getting up whatever the sap is, they're to be sap-eating insects. And so they will be taking up all of the virus particles that are in there and then go to the next plant and then inject it there. Okay, so spend a couple of minutes talking about some what I think are really neat examples of some plant viruses. But first we need to talk about some of these satellites. I mentioned this before. This is, I forget if this one's probably about 30 nucleotides in length, has this secondary structure. This also can get packaged in some of the virions. These guys, and particularly known for tobacco mosaic viruses, but also some of the cucumber mosaic viruses, this can give you a really nasty phenotype if you happen to be packaging a satellite RNA versus another virus which doesn't package a satellite RNA. Quite why this is, people think this may be mimicking some part of the ribosome or something like a ribosomal RNA or a tRNA, but it's really not entirely clear why these satellites have these particular phenotypes. They can't spread in the absence of some other virus, but they can be packaged and be moved around. And so people think about these as sort of the original selfish elements, RNA elements which could have been around from the RNA world. Again, how they would get spread around is still a really open and interesting question. But that you have these small RNAs, turns out that these particular residues here in this cucumber mosaic virus-associated satellite are absolutely critical for function. Quite what they're doing is still rather controversial. Just wanted to mention that this is a pretty active area of research in terms of plant viruses. Then I want to talk about some structural work. Again, I mentioned this right at the beginning. CCMV is cowpea-chlorotic mosaic virus, probably best known because of its structure. And basically, you've probably seen this picture in the corner of all my slides in the beginning. These at regular pH, i.e. on the outside of the cell, neutral pH, relatively low ionic strength, have this particular structure, and all you need to do is change the pH or ionic strength, and they undergo this really fascinating conformational change, which basically lets out the RNA, which is on the inside, through these threefold axes of symmetry here. So neutral pH, in fact low or high pH, and higher ionic strength, these have these really amazing and completely reversible structural changes which take place. This structural change takes place with just capsid protein assembled into each of these individual particles. So you don't even need to have RNA around. You can purify protein, put it together, you get these structures, and you can change these things back and forth. And so this turns out to be a really, really useful tool because these guys are extremely regular in terms of their shape and size, and so you can make all kinds of really cool things out of these individual viruses. We also know at high resolution what the structure of all those proteins are, exactly what's there. One of the things that's been done with these particles is putting large amounts of metals, and particularly ferromagnetic metals, on the inside of these particles, and then you can lay them down in an array. This is just a picture of one of these arrays right here. All these extremely regular particles, less than 50 nanometer size. It's really hard to get nanoparticles that are so regular relative to these sizes. You get a big distribution in terms of nanoparticles. However, if you make the nanoparticles by putting them inside a virus capsid, then they're all going to have exactly the same shape and size, which is really important for things like high-density magnetic storage. And so the idea, you could actually probably get an order of magnitude higher density storage on a hard drive by using this kind of technology. You could also use these for MRI, magnetic resonance imaging, for getting large amounts of spin labels. So these are usually gadolinium complexes, again, metal complexes that you put inside each of these individual particles. This is really important for getting high contrast. One of the big problems with medical imaging is that we're mostly water. And the difference between water with a little bit of this or water with a little bit of that is really hard to tell. Whereas if you can now use some of these metals, particularly if you can now target them to, say, a site of cancer or something else inside the body, then that's a really useful tool to say, hey, this is the kind of place that we want to be going after, say, with our radiation therapy, whatever it is a little bit later on. So these are very useful. They're also trying to use these for chemotherapy drugs. And the main people who've been driving this work are a guy who I saw last week in Paris, Mark Young, who also was my supervisor for about a year and a half at Montana State, and his collaborator, Trevor Douglas. If you're interested in more details on this, you can go and look these guys up in the literature. Yeah?
How do you get metal inside of a virus on purpose?
Yeah, so the idea is basically what you do. You remember that N-terminus that's got all those arginines that are associated with it? People know what specific metals will bind to, and very often they're going to be cysteine residues. So if you put cysteine residues at the appropriate location in each of those helices, so you've got 180 of those capsid proteins, you can put cysteines arranged in such a way that they're sitting on one side of the helix. That way you can put large amounts of metal at very specific places inside that capsid. You can do it under a high ionic strength or a low pH environment. All those metals go in. You change the pH, they'll collapse down. You can purify all those particles away from all the free metals that haven't bound, and then you can put them down on arrays, and usually what happens is then you blast away all the organics, and then you've got all of these little nanoparticles which were put down with the metals exactly where you designed them to be inside the virus capsid particle. Okay, another quick aside here, not very far away from Montana State University. We do a lot of work in my lab on hot spring environments, and one of the things you find in these hot spring environments are these really amazing plants, Dicanthelium lanugizonum, I always mispronounce it, panic grass, wide-leafed grasses. These are the things that are growing the closest to the hot springs that we're collecting our samples from. These plants can grow up to above 50 degrees Celsius. This is the temperature probe right here. And again, these are the plants that grow to these really highest temperatures, as far as we know, of any plants that are out there. It turns out that these plants can only grow at these high temperatures if they're infected with a fungus. And that particular fungus is Curvularia. And these are just some pictures of that fungus here, some fungal hyphae. These are the fungal cells, etc. And then, of course, we wouldn't be talking about this in a virology class if it weren't for a virus that infected the fungus, which infects the plant. And the really cool thing is that if you have a normal plant that's infected with a fungus, that's infected with a virus, it grows just fine at 50 degrees Celsius. If, however, you have a virus-free plant, it's really unhappy at 50 degrees Celsius. And you can make this virus-free plant much happier if you just reinfect it with virus. So this is, I think, probably the best example that I know of a good virus, i.e. that it's the infection with a virus which allows the organism, although it's multiple organisms now because you've got the fungus and the plant. A lot of people talk about this as the holobiont. So the holobiont is the organism itself plus all of the organisms that are associated with it. We're holobionts, for instance. All of our gut microbes are certainly part of us. Without our gut microbes, we'd be dead. And so that whole process. Yeah, do you have a question?
But if there's no fungus, does it still have the virus? It can't still have virus, right? Because it doesn't have fungus.
Right. So no fungus has neither the fungus nor the virus. And so it's in fact a fungal virus that infects the fungus. And it's only if that fungus is infected with the virus does it confer this heat-resistant phenotype. So yeah, this is just a cartoon of basically the same thing here. If the poor grass isn't colonized, then it does really badly. But if you have now this, and the name they called it is the Curvularia thermal tolerance virus, so CTHTV. These are double-stranded RNA viruses, which is really interesting, and how the heck they confer thermal tolerance on one of these grasses by having this fungal infection is nobody has a clue. But it turns out there are multiple different viruses here and multiple different fungi that can infect multiple different plants, and they all cause this same phenotype, that they're all resistant to these very high temperatures, and probably also desiccation and some of the other extremes that plants have to deal with. So just an amazing evolutionary process here in terms of, and of course, it's all about the viruses. So last thing I wanted to talk about is tobacco mosaic virus. This was the original virus way back when Bierjink, Ivanovsky showed that this was a filterable agent that could still cause disease. While the meeting that I was at in Paris was the Pasteur Institute, one of the things that I did while I was there is go and visit Pasteur's tomb, which is right below his lab. I'm not sure I want to be buried right next to my lab, but that's another story. But right there on the walls of the, basically tomb, which is there, has all of the things which Pasteur was involved in going through, which is really, pretty amazing. Curing rabies and pasteurization. One of the things they have in the museum, you probably all heard about the swan neck flask, so the spontaneous generation. If you boil the broth in it with this swan neck flask, then once that broth is boiled, nothing's going to grow in it. There's one that he is said to have made in the 1850s, which is still there and still sterile, and still open to air at one end. It's pretty wild. So that being said, he wasn't the first one to show that there were viruses, but worked again a lot on rabies and some of these other viruses. First ones to really come up with good vaccination protocols, pasteurization, etc. So this is the classic phenotype of tobacco mosaic virus, although all of you who are plant biologists who know much more about this than me could say, well, hey, Dr. Stedman, that's not a tobacco leaf, it's tomato. So it turns out tobacco mosaic virus does a really good job on tomato as well. So again, good explanation that the name of whatever virus it is may or may not reflect what plants it actually does infect. But again, this is the original virus and was found as these rod shapes. What's interesting is that there was a lot of argument. Now, so people found that these were filterable infectious particles, but for a long time, people actually thought that TMV was just protein, and a lot of viruses were just protein. And so we'll talk about infectious proteins a little bit later on, right, George? Some of our last classes of the term. But Wendell Stanley, who originally crystallized TMV and got crystals of TMV, and so that crystallization of TMV was basically one of the main reasons that people said, oh, viruses aren't alive because you can't crystallize life. Because of that, he got the Nobel Prize in 1948, and he was convinced that TMV was just protein. Because if you look at tobacco mosaic virus, it is mostly protein. But there's still, of course, a little bit of nucleic acid which is in there. Just wanted to add a little history here, maybe a little bit hard to see the car here in front of Stanley Hall, which was one of the main research buildings built on the Berkeley campus, basically right after Wendell Stanley, actually right before he got the Nobel Prize, moved there. I did a lab rotation in this lab up here on the corner, not driving this car, not quite that old, but this building was also rebuilt in 2007. This is what it looks like. Just again, so Stanley Hall, what people are doing, and that was basically the building that TMV built because they were all interested. It turns out that TMV, once they figured out that it had nucleic acid as well as the protein, turned out to be a wonderful source of nucleic acid in general, and particularly RNA, because it's a single-stranded RNA that was used to study translation, et cetera, because you can get huge amounts of this single RNA in terms of looking at that. And also just in terms of looking at protein structures, looking at helical symmetry. Rosalind Franklin did a huge amount of work on TMV as well in terms of X-ray diffraction. It was a really wonderful model for doing that. Just wanted to mention here, this is the structure now of TMV. This is the highest resolution structure that we have to date. The little red circle you can see in the middle there, that's the single-stranded RNA, and then all the rest of it is protein, and also gives you a pretty good idea of the length versus width of these particular particles. They're really long and very skinny. This is probably, again, the highest resolution structure that we have of any helical virus. One of the big problems, of course, with any of these helical viruses is what do you do with the ends? TMV seems to be perfectly happy with any of these ends. Turns out that TMV is so stable that you can go to pretty much any cigarette butt and isolate TMV from the tobacco which you can find in any of these cigarettes. We got some from our colleagues down at Oregon State, but he said, don't worry about it. Just find some cigarettes and you'll be able to isolate. Find some cigarettes, put some buffer on them, scrape up the leaf of a susceptible tomato. Most of the tomatoes, in fact, are resistant. And then you can get infection. We didn't end up doing that, but that was the possibility and how you could do that. So these things are ridiculously stable. So that's all I have for plant viruses. Again, unfortunately, I apologize to any botanists and plant virologists out there. This is very much an overview of that process. So I will also have clicker questions again on adenovirus when we meet again, which will be Friday. See you then.